Free DNA analysis for non-invasive examination of trisomy. Use of cell-free DNA screen for fetal aneuploid with the use of cell-free DNA obtained from maternal plasma and have detection rate of trisomy 21 syndrome Down.
And genetic insight into a debilitating condition.
The connections are not correct, and the processes aren't correct, and there's some sort of deficit.
The directions for a human being are written in code, 3 billion letters long. These instructions tell our bodies how to live, how to grow, how to die. Researchers call this code the sequence.
Just as twins can be the result of unusual circumstances at conception or in early development, so can many kinds of birth defects. Early errors in the number or structure of chromosomes can lead to birth defects like Down syndrome.
About Down syndrome, the human body is made up of cells. Normally, the nucleus of each cell contains 23 pairs of chromosomes, for a total of 46. In babies born with Down syndrome, each cell contains an extra chromosome, chromosome number 21, for a total of 47 chromosomes in each cell. This condition is usually caused by an error in cell division in the earliest stages of development.
Almost 50 years ago, early genetic researchers discovered the presence of this additional chromosome in people with Down syndrome. They assumed that it alone caused the physical and developmental disabilities shared by people with Down. But today, our increasing ability to look at specific genes within each chromosome is giving scientists a clearer picture of what causes the condition. About 1 in 800 to 1,000 births results in a child with Down syndrome. It is
Study population and sample collection. Illinois patients were at least 18 years of age and had a sampling premising between 10.0 and 14.3 weeks of gestation at the time of study blood sample collection. Gestational age was determined according to the ground room lead at the time of the measurement of neutral translation, permanence, and neuron outcomes. In a blinding fashion, the first and last authors reviewed medical records of all neonates with congenital anomalies and excluded those with phenotypes suggestive of aneuploidy. Of new confirmatory genetic testing was performed. Results of fetal and newborn genetic testing were adjusted to clinical genetics, categorized as euploid or aneuploid, and classified according to the type of abnormality.
Down syndrome have physical and cognitive disabilities ranging from mild to severe. They are able to lead full lives with good medical care and education.
Good morning, Elson. How are you? Good. Good. How are you doing? Not too bad.
Individuals with Down syndrome can often have lifespans into their 50s, and perhaps more. The aging process, though, is somewhat accelerated. Individuals with Down syndrome are at much higher risk for having an Alzheimer-like condition develop in their central nervous system, in their brains. And this leads to a decline in some of abilities that may begin as early as their 20s or 30s.
So what is it about about this extra chromosome that affects the brain of people with Down syndrome. Scientists at the University of Wisconsin in Madison are using the latest tools in genetic research to look at each gene to better understand what happens in development and why.
We started a study looking at post-mortem fetal tissue collected from Down syndrome patients, where we knew that the fetal tissue had Down syndrome. And we isolated stem cells from that fetal tissue. We asked the question, are there any genetic differences in the human stem cells grown from Down syndrome when compared to non-Down syndrome stem cells?
The use of fetal stem cell research is controversial, but it's giving scientists an unprecedented window into early human development. Before human stem cells were available, the only way scientists could look at the genes that cause Down syndrome was by studying mice. But mice don't.
Result: among the fighting beyond A41 permanents in the primary analysis population, there were 68 chromosomal anomalies, 1 in 236 permanents. Of these anomalies, 38 were trisomy 21, 10 were trisomy 18, 6 were trisomy 13. Trisomy 21 was identified of 38 of women for a prevalence of 1 in 14,170.
Cara futura mamá, don't be afraid. Tu niño podrá hacer muchas cosas.
Podrá marcharte. Podrá correr hacia ti.
Il pourra parler et étudier l'Italie. Il pourra aller à l'école como todos. Il pourra t'écrire si un jour elle est loin. Because indeed, he'll be able to travel too.
Il pourra aider son père à réparer son vélo.
Podrá laborar y ganar su dinero.
Avec son salaire, il pourra t'inviter à dîner.
O affittare un appartamento e andare a vivere da solo. A volte sarà difficile. Jak ciężko.
Quoi de possible? N'est-ce pas pareil pour toutes les mères?
Dear future mom. Tuo figlio potrà essere felice. Come lo sono io.
Et tu seras heureuse aussi.
Vero mamma. Vero mamma.
Conclusion: the performance of DNA testing was superior to that of traditional first trimester screening for the detection of trisomy 21 in a return prenatal population, although these data support the use of DNA testing in women regardless of age or risk status. For cause-utility studies were obtained and implemented by professional societies. 26 of useful DNA testing of other genetic tests requires of explanation of the limitations and benefits of prenatal tests shows the pattern.